Hi everyone, this is Shakil Ahmed, your biology educator. And today we will discuss another MCQs paper, Summer 23, Variant 13. And we will select I have actually chosen this paper because we have the paper 1 exam coming up in the next week. So it will be a good practice for the students to go through these papers. So let's start from question number 1 over here. Which feature is visible with a light microscope using a natural light source? We know the natural light source, it has a minimum wavelength of 400 nanometers. And so resolution will be half of 400 nanometers is 200 nanometers. So any object which is more than or equal to 200 nanometers in size will be seen using the natural light source in a microscope. So DNA molecule of diameter 2 nanometers is not possible because 2 nanometers is much less than 200 nanometers. Paramecium cell of diameter 200 micrometers. So because we know 1 micrometer is 1000 nanometers. This means the size of the paramecium cell is 200 into 1000. So it's much bigger than 200 nanometers. Phospholipid bilayer of width 8 nanometers, again less than 200 nanometers. A ribosome of diameter 20 nanometers. So that means option B is the correct answer. Moving to question number 2. The electron micrograph shows a structure found in the cytoplasm of an animal cell. Which biological molecules are found in this structure? So if we look at this photograph minutely, we'll see that there are present 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 actually structures are making a circular structure, which is actually, we know it's a centriole. And centriole is the structure present in the animal cells, which has the 9 microtubule, those doublets surrounding it. So which biological molecules are found in this structure? Nucleic acids, no. These are completely the microtubules, and microtubules are made up of proteins. Proteins is yes, and they don't contain any phospholipids. So option D is the correct one. Question number 3, which cell structures contain nucleic acids? Cytoplasm, yeah, it contains the nucleic acids. Because cytoplasm will have the cell organelles in it. Lysosomes, they don't contain any nucleic acid, they have nothing to do with the nucleic acids. And mitochondria, we know the mitochondria have the nucleic acid inside them. So it is 1 and 3, that is option C is the correct answer. Mitochondria are thought to have evolved from prokaryotic cells that were ingested by an ancestral cell. Which feature have prokaryotes lost during their evolution into mitochondria? We know that all prokaryotes, they contain a cell wall. A typical structure of feature of the prokaryotes is that all prokaryotes contain a cell wall around them. But when we see them in the mitochondria, there is no cell wall. So the feature that they have lost during the evolution is the cell wall. Circular chromosome, it's still there. We're not having any endoplasmic reticulum because these are the prokaryotes do not have membrane-bound organelles. And ribosomes, they are still present inside them in the mitochondria. Question number 5, which polymers are present in all viruses, all prokaryotes and all eukaryotes? Polynucleotides means the nucleic acids, whether it is DNA or RNA, it's present in all the viruses, prokaryotes as well as the eukaryotes. Polypeptides means the proteins. Proteins are also present in all viruses, prokaryotes and because viruses have the membrane of the covering capsid made of the proteins. Polysaccharides are the sugars. We may not have sugars in viruses, and we may not have sugars in some prokaryotes as well. So that means option 1 and 2 is the correct one. D is the correct answer. How many of the listed structures typically contain genetic material that has telomeres? So telomeres are present in the chromosomes, and especially they have a role to play in the cell division, mitosis, to prevent the loss of the ends. So those cells that undergo mitosis will obviously have telomeres in there with the DNA. So bacterial cells, they don't undergo mitosis. Chloroplast, no. Mitochondria, they don't have the mitosis. Yes, nucleus is the one which contains chromosomes inside it. So it is just one structure, nucleus. Steps 1, 2, 3 and 4 are used to test for a non-reducing sugar. Put 5 centimeter cube of solution into a test tube. It's a non-reducing sugar, and we know in a non-reducing sugar test, we first use the solution, put the hydrochloric acid in it, and then heat it. And when it is heated, so that we do the acid hydrolysis of the non-reducing sugar. Then we add some alkali to neutralize any acid, and then we add the Benedict solution and heat it again, because we have changed the non-reducing sugar into the reducing sugar. So the first step is put 5 centimeter cube of the solution into a test tube, that's correct. Add a few drops of acid, that's correct, and neutralize. So first of all, we heat it over here. Then we allow it to cool and neutralize it with alkali, add 6 centimeter of Benedict solution, and then after that we heat it again to see if there is any color change. That means it is after 1 and 2, between steps 2 and 3, and after step 4. B is the correct option. Moving to question number 8. Which features contribute to the function of a cellulose molecule? Long chains of glucose molecules coil into a helix. We have not seen any helical structure formed in the cellulose. So this option is wrong. Many hydrogen bonds form between adjacent chains. That's correct, because in a cellulose we know that every second beta glucose molecule is turned upside down, and the hydrogen bond is formed between carbon number 1 and carbon number 4. And it is insoluble in water. That is also correct, because if cellulose is soluble, then it will not be of any use. So that means options 2 and 3 means C is the correct answer. Question number 9. What correctly describes triglycerides? We know triglycerides are the same as the lipids, and lipids are insoluble in water and soluble in the alcohol. And all the triglycerides, if any one of them is not replaced by phosphate, they are non-polar molecules. So they are non-polar that are soluble in ethanol, that's the correct one. Non-polar that are soluble in water, they are not soluble in water. Polar, they are not polar molecules, they are not polar molecules. So option A is the correct one. Question number 10. Cocoa butter contains 3 different triglycerides. These triglycerides are made from the fatty acids. Oleic acid is O, palmitic acid is P, and stearic acid is S. The 3 triglycerides found in the cocoa butter are POS, SOS, and POP. The chemical structure of the triglyceride POS is shown next to a diagrammatic representation of POS. So we have, this is P, this is O, and this is S. Now what is the question? Which statement is correct? Triglycerides POP contains 2 unsaturated fatty acids joined by ester bonds. Do we see 2 ends? No, there is only 1 unsaturated fatty acid, because the unsaturated fatty acids contain a double bond. That means option 1 is not the correct answer. Triglyceride POP contains 2 less carbon atoms than triglyceride POS contains 2 less carbon atoms than triglyceride POP. Let's see first of all what's the difference. If this is P, we have, I'll count from here the number of the carbon atoms in without the ester bond. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So this one contains 15. So this one will be 16, 17 with oleic acid. And again this one is 17. So S also contains 17. So POS means 15 plus 17 plus 17, which means 17 plus 17 is 34, 39, 49. And POP means P was 15. So this means 15 plus O is 17, again 15, which means 47. So there is a difference. So triglyceride POS contains 2 less, no, it contains 2 more. So that means this option is also not correct. Triglyceride SOS contains 4 more carbon atoms than POP. We have already found POP, which is 47. Let's see about SOS. S contains 17, O contains 17, and S contains 17. That means 17 3s are 51. So SOS contains 4. 51 minus 47 is 4. So that is the correct answer. Now let's look at the option D as well. Triglyceride SOS contains 2 saturated fatty acids joined by the hydrolysis to glycerol. So they are not joined by hydrolysis, they are joined by the condensation reaction. Which molecules contain at least 4 double bonds? It's at least 4 double bonds. Unsaturated triglycerides. Unsaturated triglycerides means if this is the glycerol, then we have the 3 fatty acids and they contain the double bond. So we have 3 double bonds here. And if they are unsaturated, means there will be some, even if one of them contains a double bond, it's unsaturated, that means yes, it contains. Hemoglobin is a globular protein with a quaternary structure made up of 4 polypeptide chains. And we know in the hemoglobin also we have the double bond present in the proteins. So because proteins contain the acid C double bond O and NH. So this also contains the double bonds. And collagen is also a protein, obviously this will also contain, so all of them contain at least 4 double bonds, so D is the correct answer. Because collagen molecule is made up of 3 polypeptide chains first of all. So we have all of them contain the double bonds between them. The diagram shows the amino acid, glutamic acid. What is the R group for glutamic acid? Now we know the amino acids contain the parts like we have carbon atom. Each amino acid has the COH acid group with it. Each amino acid has the amino group NH2 with it. Each amino acid has hydrogen with it. And then the one which is different in the different amino acids is the R group. So we have COH is here, so this one is gone. NH2 is here, so this one is also gone. Then hydrogen is here, that means this is CH2, CH2, COH. D is the correct option. Now the diagram shows 3 interactions that hold protein molecules in shape. Which row identifies these interactions? So we can see the interaction 1 is there is a small negative charge on the oxygen, small positive charge on the hydrogen. The force attraction between them we call it as the hydrogen bond. There is S bond, this is a disulfide bond, or disulfide bond is formed by the sharing of the electrons, and the sharing of the electrons is the covalent bond. And then there is NH3CO, which is one is positive and other is negative. The interaction between the oppositely charged molecules, where we have the one is fully positive and negative, that is what we call the ionic bond. So interaction 1 is hydrogen, 2 is covalent or disulfide, and 3 is the ionic. Hydrogen, covalent and ionic. So we cannot select A because it shows here hydrophobic interaction, and this one in interaction 3 is not a hydrophobic interaction. Question number 14. Some animals produce antimicrobial proteins which protect them from pathogens. These proteins could be used to kill human pathogens. However, when used as a medicine, they are broken down by protein digesting enzymes. Replacing one of the amino acids found in the protein with an amino acid that had been synthesized in the laboratory resulted in a modified protein that was not broken down. What could explain why this modified protein was not broken down by the protein digesting enzyme? The modified protein has a different tertiary structure to the original protein. This could be the reason. If the structure changes, it will not fit into the active site of the enzyme. The modified protein is not complementary to the shape of enzyme. The same thing there. If it is not complementary to the shape of the active site, it cannot fit. So enzyme will not be able to break it down. The modified protein is unable to induce a fit with a protein digesting enzyme. Again, the same thing. This is correct one, because if it is not able to induce fit mechanism, then it will not be broken down by the enzyme. Then all the 3 options are correct, 1, 2 and 3. Moving to question number 15. A student investigated the effect of substrate concentration on the rate of an enzyme catalyzed reaction. A graph was plotted to show the relationship between 2 variables. The student was asked to take readings from the graph that could be used to determine the Mitchell-Menton constant Km for this enzyme. J, K, L and M show points read from the graphs which the students could use to determine the values. We know J is the Vmax. K is where we find the substrate concentration for the Mitchell-Menton constant. And L is the half of Vmax. And M is the substrate concentration when it is having the maximum Vmax. Which 2 readings must the student use to determine the value of Km? So we must know where is the half of Vmax, and then we will find the value of the substrate concentration. That means L and K. So K and L or L and K, C is the correct option. Moving to question number 16. The end product of a metabolic pathway can act as a competitive inhibitor. This is called end product inhibition and allows a cell to control a metabolic pathway. The diagram shows a metabolic pathway where the end product could act as an inhibitor of the enzyme W. So if this is the end product here, so it acts as an inhibitor for the enzyme W. So as long as when the concentration of the end product increases, so enzyme W will not be able to work. So that means the intermediate one will not be produced, so it will stop. It will work that way. Inhibition is actually this is what we call the end product inhibition. What would be the effect if the enzyme Z was inhibited by the end? Now, instead of enzyme X, if they say enzyme Z was inhibited, that means now enzyme X will not have the inhibition. So we will see that the intermediate 2 will increase. And because now it is inhibiting enzyme Z, so less of the end product will be produced. So quantity of intermediate A will increase, so we have one of these, but the quantity of end product will decrease, so A is the correct option. Question number 17. Which row shows the distribution of cholesterol and the carbohydrate chains of glycolipids and glycoproteins in a cell surface membrane? So cholesterol molecules, we know if this is a phospholipid, then this is another phospholipid, this is another phospholipid. We have the bilayers between them, we have the bilayer. The cholesterol molecules fit themselves between them. So we can see the cholesterol molecules are present between the phospholipids because cholesterol molecules will also have the hydrophilic phosphate end and then the hydrophobic tails. Carbohydrate chains of glycolipids and carbohydrate chains of glycoproteins. So we have to tell where are these chains present. These chains are mostly present on the glycoproteins on the outside. Even on the phospholipid, they are present on the outside. So we have cholesterol between the phosphate molecules, so one of these. Inner on the inner surface, no, it's on the outer surface, D is the correct option. Question number 18. Which row correctly describes all the possible relative concentrations of a substance when the substance moved by the endocytosis or exocytosis? We know both endocytosis and exocytosis. They are the active, they take place by the active transport or the bulk transport is there. So active transport always involves the movement against the concentration gradient. So what we see is that endocytosis, concentrations are equal. No, we cannot say that there is the concentration gradient also. So concentrations equal greater inside or greater outside, concentration equal greater inside or this is the one that is the correct option because whenever there is the concentration gradient, we see the active transport can take place. This statement about simple diffusion is correct. It requires a specific molecule in the cell surface membrane that's for the facilitated diffusion, not for the simple diffusion. It's a passive mode of transport of substance. Yes, exactly. This is correct because diffusion does not need any energy. It always requires a membrane, so membrane is needed for the osmosis. It only happens in the cells of prokaryotes and eukaryotes. No, diffusion even takes place outside the cells as well. So it is passive mode of transporting the substance. Question number 20. Plant cells with the same water potential in their cytoplasm were each put into one of three different concentrations of sugar solution. So the concentration of the sugar solution 10%, 5% and 2.5%. The cells were left for 50 minutes and then observed using a light microscope. We can see the cell X has not lost water, it has almost remained same because the vacuole is small. Cell Y has gained water. We can see the vacuole has increased in size, the cell is stirred, and cell Z has undergone plasmolysis, it has lost water. Which statements are correct? Cell Y had a lower water potential than the sugar solution it was put in. Cell Y had a lower water potential. This is correct because if it has a lower water potential, then only the water will enter in the cell Y. Cell Z was put into 10% sugar solution. Z was put in 10% exactly because here the plasmolysis, the highest concentration is. This is also correct. Cell Z had a less negative water potential than the sugar solution it was put in. Cell Z had a negative. Less negative means higher water potential. More negative means lesser water. So cell Z had a less negative water, means higher than the sugar solution it was put in. That's correct. That's why it has lost water. So all the three options, 1, 2 and 3 are correct. So A is the correct answer. Which row describes some properties of stem cells? They are able to divide by mitosis. This is actually what we mean by the stem cells. When they divide, then they differentiate into the specialized cells, and they are able to repair damaged cells. They don't repair damaged cells, they repair the damaged tissues. Cell cannot be repaired, cell can be replaced. So we see that first two are correct and third one is wrong. B is the correct option. Moving to question number 22. The mitotic index is a measure of the proportion of cells that are undergoing mitosis in an area of tissue. It is calculated using the formula. Mitotic index is number of cells undergoing mitosis divided by total number of the cells multiplied by 100. This will give us a percentage. So a scientist calculated the mitotic index of the areas of onion root at different distances from the tip of the root. The results are shown. So these are the distance from the tip, 0.2 millimeters, 0.4, 0.6, and this is the mitotic index. So we can see the mitotic index is highest when we have the 0.2 millimeter distance, and then it keeps on decreasing faster, then it goes. So what is the question? Which statement is correct? No cell division occurs further than 1.4 mm from the tip of the root. So it is here, it's still taking place for the 1.4 mm, but it remains constant. Means the number of cells undergoing the mitosis, all the cells are undergoing mitosis, is remaining same. The rate of cell division decreases as the distance from the root tip decreases. As we see, the rate of mitosis index is opposite. It decreases as the distance from the root tip increases. So this is also the wrong option. Most of the cells undergoing cell division are closer to the tip of the root. So we have at the 0.2 mm distance, the cells have the highest mitotic index, then it goes on. So this seems to be the correct option. For a sample of 200 cells, 0.2 mm from the tip of the root, 6 would be undergoing mitosis. So how can we have 6 by 200 cannot be the mitotic index of 12? So this is wrong. 6 by 200 into this is, it has a mitotic index of 3 only, not 12. So that means D is also the wrong option. Which molecules make up the structure of ATP? ATP, we know it has the ribose sugar, it has the adenine, and then it has the 3 phosphate molecules with it. So it has adenine, yeah, because ATP is adenosine triphosphate. Thymine is not there, deoxyribose is not there, phosphate is there, there are 3 phosphate molecules and ribose. So 1, 4 and 5 means C is the correct option. Moving to question number 24. A short piece of DNA, 19 base pairs long, was analyzed to find the number of nucleotide bases in each of the polynucleotide strands. Some of the results are shown. How many cytosines were in the strand 1? So let's first of all look at the, this is 19 base pairs long. So if we have 4 thymine here, in the strand 2 there will be 4 adenine, because thymine always bonds with adenine. If we have 5 thymine here, so we will have 5 adenine in the strand 1. This is total 19. So 4 plus 7 is 11, 11 plus 5 is 16. That means 3 are the guanine. Guanine always bonds with the cytosine. That means the strand 1 will have 3 cytosine molecules. So B is the correct answer. Which statement about the role of DNA polymerase in the process of semi-conservative replication of DNA is correct? DNA polymerase forms the hydrogen bonds between complementary base pairs, which is wrong. It also forms the hydrogen bonds between the incoming nucleotides. DNA polymerase, it forms the phosphate bonds between them. DNA polymerase moves along the lagging in the 3' to 5'. No, it moves in the opposite, 5' to 3'. DNA polymerase joins new bases to the leading strand only. No, it joins new bases to the lagging strand as well. DNA polymerase moves along leading and lagging strand in the 5' to 3'. This is correct one. That's why we have the Okazaki fragments formed in the lagging strand. The diagram shows the process of translation occurring at a ribosome. What is the base sequence at S? S is over here. This is anticodon. So if we have C here, it should contain G. If we have U here, it should contain A. And if we have A here, it should contain U, because we don't have T in the RNAs. So GAU means C is the correct option. Question number 27. Which row correctly explains how the structure of phloem sieve tube elements and xylem vessel elements are related to their function at transport system? Contain mitochondria to make ATP. They don't contain mitochondria for the loading of sucrose. So actually mitochondria present in the companion cell, not in the phloem sieve tube. Cell walls are thickened and contain lignin, which makes xylems transport waterproof. Okay, so this option is wrong here, so this cannot be the answer. Contains no nucleus, so that dissolved solutes can move easily through cells. This is correct. End walls between the cells are perforated. We don't have end walls in the xylem vessel elements. End walls between the cells are perforated, so that dissolved solutes can move between the cells. This is correct. Lignin in the cell walls gives strength so that xylem vessels do not collapse due to transpiration pool. This is also correct. So option C is the correct one. No end walls between the cells, though this is wrong. Contains no cycle, that's correct, but no. Now question number 28. Which component of plants is used by the apoplast pathway as water is moved from the soil to the xylem? So the xylem. Caspian strip is not used in the it prevents the apoplast pathway. Cellulose is correct. Endodermis, no, we use it. And suberin also is the waterproof. It prevents the movement of water. So B, cellulose is the correct answer. Question number 29. Which statements correctly describe transport pathways in dicotyledonous plants? In the simplest pathway, water may move through the intercellular spaces. This is correct. The simplest pathway may be blocked by the tonoplast, which is wrong. In the apoplast pathway, water does not move through the plasmodesmata. This is correct. The apoplast pathway may be blocked by the Caspian strip. This is correct. So we see that in the water may move through the intercellular spaces. This is actually not correct. So in the plus, water does not move through the plasmodesmata. So D is the correct option. Question number 30. Plants such as tobacco plant retain very little of the water they take in, and the volume of water lost during transpiration is very high. This is because these plants can only absorb carbon dioxide through open stomata. Plants use carbon dioxide to synthesize glucose molecules. It's estimated that 400 molecules of water are lost for each carbon dioxide molecule gained. How many water molecules are lost for a plant to synthesize 1 molecule of glucose? So we know if the amount of the water and the carbon dioxide, 6 times carbon dioxide, 6 times water, it gives us the glucose and 6 times oxygen. So if we have 400 molecules being used in moving 1 carbon dioxide molecule, that means 400 into 6 will be, because 6 are needed, 2400. So then 6 carbon dioxide will fuse with the 6 water molecules. That means 2400 is the correct answer. 31. Which changes occur as sucrose is transferred from leaves into the phloem sieve tubes to be transported to a sink? Water potential in the phloem sieve tube becomes, when water potential becomes more negative, then only the water will move into them. And volume of liquid in the phloem sieve tube, when it is more negative, means less water potential, the water will enter them, so the water molecule will increase. So more negative and increase is the correct answer. Question number 32. The graph shown does not have any axis label. Which row shows appropriate labels for the axis that would explain mass flow in the phloem? If x-axis is distance from the sink, so distance from the sink increases, hydrostatic pressure decreases. That's wrong. As we see, the distance towards the sink decreases, hydrostatic pressure decreases. Hydrostatic pressure on the x-axis and distance from the sink, no, that's also not, because if we decrease the distance, the hydrostatic pressure will not decrease. Distance from the source, M, and hydrostatic pressure, K. So if we increase the distance from the source, we'll see the hydrostatic pressure goes on decreasing. That is the correct one. What is present in the blood, in the human veins? Chloride ions, carbonic anhydrase, oxyhemoglobin. Chloride ion may be present because we know that blood contains carbonic anhydrase and it contains the oxygen. Not all the hemoglobin molecules lose oxygen. So all of these options are correct. So 1, 2, and 3 is the correct. A is the correct option.